Beta oxidation of unsaturated fatty acids. First, the monounsaturated fatty acid. The pathway of beta oxidation of unsaturated fatty acids is same as that of saturated fatty acids until the double bond is reached. For example, in palmitoleic acid, a 16 carbon chain fatty acid which has a double bond at 9-10 position, after 3 cycles of beta oxidation, cis delta 3 enoyl coenzyme A is produced. This molecule is not a substrate for the enzyme acyl coenzyme A dehydrogenase, which catalyzes the first step, that is oxidation of the beta oxidation of saturated fatty acids, and also produce a molecule of FADH2 in the process. Therefore, another enzyme, enoyl coenzyme A isomerase, first rearranges the cis delta 3 double bond to a trans delta 2 double bond, which then proceeds normally through the usual beta oxidation pathway. Thus, the enzyme isomerase catalyzes the cis trans isomerization and transfer of double bond position. This enzyme is therefore very important because, unlike trans double bonds introduced during beta oxidation, the double bonds of most naturally occurring unsaturated fatty acids have a cis configuration. The energy yield from an unsaturated fatty acid is less as compared to saturated fatty acids. As we have earlier seen, one energy yielding step is bypassed in the process during the oxidation of unsaturated fatty acids, that is, the first dehydrogenation reaction catalyzed by the enzyme acyl coenzyme A dehydrogenase, in which a molecule of FADH2 is formed. Now, beta oxidation of polyunsaturated fatty acids, for example, linoleic acid, which has 2 double bonds, that is cis delta 9 and cis delta 12 double bond, till the first double bond, the molecule is oxidized like monounsaturated fatty acid. Problem arises in the 5th round of the beta oxidation, which results in the formation of 2,4-di-enoyl coenzyme A. This molecule is a poor substrate for enoyl coenzyme A hydratase, and therefore this problem is solved by an enzyme 2,4-di-enoyl coenzyme A reductase. This enzyme reduces the double bond and produces trans 3-enoyl coenzyme A. Trans 3-enoyl coenzyme A must first be isomerized to trans 2-enoyl coenzyme A, and this is carried out by the enzyme 3,2-enoyl coenzyme A isomerase. After this, the process proceeds along the beta oxidation pathway. Beta oxidation also occurs in peroxisomes apart from mitochondria in animals, and in plants and most fungi it occurs only in the peroxisomes. Peroxisomal beta oxidation is similar to but not identical with the mitochondrial beta oxidation. Enzymes are different, but the chemical transformations of the fatty acids are the same in both. Peroxisomal beta oxidation does not degrade fatty acids completely but acts as a chain shortening system, catalyzing only a limited number of beta oxidation cycles. Therefore, majority of long fatty acids, that is those having more than 20 carbons, are metabolized by beta oxidation first in the peroxisomes and then in mitochondria. FADH2 and NADH produced during peroxisomal beta oxidation is not coupled to oxidative phosphorylation. The electrons from FADH2 in the peroxisomes are directly transferred to oxygen, which is reduced to hydrogen peroxide. Since it is not coupled to oxidative phosphorylation, therefore no ATP synthesis takes place and it generates heat. The NADH formed cannot be reoxidized in the peroxisome, so it is exported to the cytosol and eventually to the mitochondria.